Okay, welcome to this course, Algorithmic Thinking in Bioinformatics. So, in this course, the main idea of this course is to understand where and how to apply different algorithms and ML models in real life applications, especially in biological applications, like for to address biological questions. That will be the basic idea of this course. Mostly, we will look into the different types of algorithms and what kind of algorithms and can be applied to solve biological problems and how to apply these algorithms. But in few weeks, we will look into different ML models also. The second idea for this is to apply these algorithmic approaches to address these valuable questions and to concern these biomedicine related problems. Okay? So this is the structure of the course. So every week we will start with a biological question. This will how the entire course will be structured. Every week we will be addressing one particular biological question. So we will start with a biological question and then we will reframe it as a computational problem. Okay. So now in biological question, every biological question will have some biological inputs also like a gene or a DNA sequence or whatever it is. So we will have some inputs that will be real life inputs, or obviously here we will be simulating these inputs. But in real life, we will start with a biological question that will require some biological input, and we want that the problem should give us some kind of an output. Okay, so we will reframe this biological question in the form of a computational problem. So now, as you already know from the PDSA course, that the computational problems may or may not be solved. There are P problems, there are NP problems also, right? So we may be able to solve some problems. There may be polynomial time algorithms to solve some problems, and for some problems, it may be the case that we do not have any polynomial algorithms, or it may also be possible that these problems are still an open question. We don't know whether it can be solved in polynomial time or NP time. Okay, so it also may be possible that few sections of this computational problem, like this entire computational problem, we may not be able to address it entirely, but some parts of this computational problem may be solvable polynomially in polynomial time, and it some parts may still be unsolvable. Okay, so whatever segments of this computational problem we can solve, okay, we will look into those portions. We will look into whether the entire problem can be solved. We will look into what problems related to this problem can be solved, what parts of this problem can be solved, and what we can do with it. Okay, so we look into these solvable portions of the computational problem, and obviously because it is solvable, there must be some algorithm to solve it, right? So we look into the algorithm. How do we design this algorithm? This black box here is assuming that there is an algorithm. So obviously there is an algorithm since we are claiming this problem is solvable. We don't know that algorithm. Okay? So we look into how do we design that algorithm. We can look into how to solve the problem, how to design this algorithm, and we look into how we can improve the time complexity of the algorithm in some segments. Okay, and we can also look into different related problems. If we can modify this problem, what kind of will it be able to help to solve the biological question to some extent and stuff like that. Okay, we'll explore this algorithm a lot, and finally, what are the outputs that whether this output is exactly equal to what we expected, right? What is the expected output when we want to solve the biological question? Does it solve the biological question or not? So if it doesn't solve, then what are the unsolvable what are the unsolvable portions of this computational problem? What are the different open problems? We look that part into very few at the last part of the week. Okay, most of the week, little bit of the first portion of the week will be based on this biological question, and to understand these inputs of the what are the inputs, the biological inputs of this for this biological question, and what is the computational problem. We'll give an introduction to the biological computational problem, and then most of the week, the major portion of the week will be based on this portion alone. like how do we solve the problem, what are the different algorithms, and how to improve upon the algorithms, what were like how the algorithm works on these biological inputs and all of that. Then at the end, we will look into little bit of if there are any open questions and if the algorithm works differently with some real life inputs, or if the algorithm has some kind of some part of the problem is unsolvable, then what are the open questions, how the algorithm works in real life inputs, what are the major problems that we face, and all of that. Okay, so that will be a small segment at the last of the week. So this will be how the entire week content will be based on. Okay, so these are the books actually. So first of all, we'll look into the major portion uh, of the entire course will be based on this book, Bioinformatics Algorithms, both the volumes, Volume 1 and Volume 2 by Philip Compo and Pavel Terzna. So this will be the book that we will be suggesting you to refer to study properly. So most of the quizzes will be based on this question on this book. Most of the content is based on this book and everything. So this is a reference book for Dan Gusfeld, Algorithms on Strings, Trees, and Sequences. This is a reference book, but for few of the weeks only. It's not for all the weeks. And in some weeks, we may require to read some new concepts which are not incorporated in any books. So they are there are some few papers on that. So we will give the references with the due course of time. Okay, in that particular week, whichever for whatever week's content it will be relevant, we will give the reference for that particular week during that week. Okay. So this is the evaluation structure. There will be weekly assignments. There will be programming assignments for few of the weeks. And this is actually a link. It's called rosalind.info. If you go to this link, there are several practice assignments for this course. So
Ma'am, is
for the
this link provided to us in the portal? The practice
I
link?
believe not so. I believe not so. We'll share the link. Okay.
Okay, ma'am.
So this link is actually it has some practice problems for the bioinformatics course. Okay, so like whatever problems you will be facing in the programming assignment, some related problems. These are for practice problems. You can look for those problems in this particular link. Okay, this entire website is based for this practice problems on the for the programming assignments on this algorithmic thinking bioinformatics course. So there will be quiz one, which will be based on content from week 1 to week 4. Quiz 2 will be based on contents from week 5 to week 8. And then the end term will be for all the weeks, but more weightage will be on week 9 to week 12. Okay. This is a brief idea of week 1, whatever we have covered in the week 1, a summary of that. So in week 1, we have looked into gene, DNA, concepts on what is a gene, what's a DNA, what's an RNA, what are these amino acids and proteins and all of that. Then this is a brief idea on how it looks. Our body is composed of cells, and each cell has a nucleus within it. Inside the nucleus, there are these multiple chromosomes, and when you expand those chromosomes, every chromosome is is basically a compressed structure of the entire DNA. So the DNA then converts into a from the DNA we get the protein RNA, and from RNA we get a protein. Okay, so this entire process of generating the proteins from the DNA is called as a central dogma principle. So we have a DNA which undergoes a process called transcription and generates the mRNA. mRNA undergoes a process called translation to generate the protein. Okay, so that is the idea behind the central dogma principle. So protein is basically composed of these amino acids. There are multiple amino acids that stick together, and one amino acid forms a protein. It's something similar to magnets. You have the small magnet balls, and one magnet ball comes close to another, and they keep on attaching itself to form a chain, and then when you release it, they just compress itself and combine themselves together, and they form a complex structure. That is how a protein looks like. Okay, so this is how DNA, the concept of a DNA. So the structure of our DNA. So a DNA is basically a double helix structure. It means that you have one spiral and another spiral entangled together. So that is how a DNA looks like. So there are two springs. You join the two springs. Whatever you get, that will be how the DNA will look like. Okay. So this what happens is in the DNA we have, if we open this DNA, no, this is something how it will look like, and there is a, these are basically chemical compounds. So there is a phosphate, basically a phosphate-like structure connected to a sugar base, and then there will be a nucleotide. Okay. So each of these base pairs are called as nucleotides. Each of this one, there are four different types of base pairs: ATGC, adenine, thymine, guanine and cytosine. A and T combine together, G and C can combine together. A and G, A and C, T and C, T and G. Any other combination is not allowed. Only A and T and G and C can combine together. Okay. So what I mean is here on one side, if there is an A, then on the exact opposite opposite side of the helix. Okay. Those two, that can be only a T. It cannot be anything else. Okay. So we have a sugar, a phosphate, and then each one of these nucleotides, and these nucleotide, sorry, each one of these ATGC combinations, and each of these ATGC one, each of these compounds are called as, these nitrogenous bases are called as nucleotide. Okay, so these are called nitrogenous bases because they are consisting of, they're made from this nitrogen. Nitrogen forms the major compound, major element in this compound. Okay, so these are, that's why they are called nucleotide. Sorry, that's why they are called nitrogenous bases, and each of these is called a nucleotide. So this is how a DNA looks like. Now, if you open one of these DNAs, take just one side of the DNA and you replace this T with what is called as an uracil. Okay, the T is basically replaced with uracil, U. Okay, then that is a RNA. That's exactly what an RNA looks like. More specifically, that's what an mRNA looks like. It's called a messenger RNA. The role of this messenger RNA is basically self-explanatory. It transfers it transfers some information. Okay, that's why it's called a messenger RNA. So this is how an RNA looks like. tRNA or transfer RNA is something different. It basically consists of exactly three uh, nucleotide bases. This will be exactly the same how an RNA looks like, but instead of a long string of these nitrogenous bases or nucleotides, there will be exactly three nitrogenous bases. And at the one end, there will be three nitrogenous bases, but at the other end, there will be one amino acid. Okay. So these amino acids, so one tRNA or this transfer RNA will, these tRNAs will combine together. So each of these amino acids, these amino acids will act like magnet balls. And when these tRNAs come together, no, these mRNAs will stick to each other. Okay. Sorry, these amino acids will stick to each other. And once this thing is done, when the tRNA stop coming together, when the stop is reached, okay, at that time the amino acids, the string of amino acids will be released, and they'll just compress itself, combine itself, they just entangle itself, and it will form a protein. Okay, so that is actually the process of central dogma concept, which I'll come to next here. So in transcription, what happens is the DNA, it will unfold. We have this two helix structure, the double helix structure, or two strings, if you can two springs, if you can imagine that way easily. So they are combined together. You open a part of it. Okay, so just one segment of it, you open it. It will look something like this, and then one something called RNA polymerase will come and stick itself to one portion. This idea of this RNA polymerase is to basically hold this opening together. It act like a wedge to open it. Okay. And it will also act like a starting point. This is the point where you start. Okay. So you open this RNA polymerase, and whatever is the complement of these nucleotides. So A's complement is T, T's complement is A. So complement basically means what is the opposite, what nucleotide can stick to this particular nucleotide. That's the complement of this nucleotide. So A's complement is T because only T can stick to A. So that's why whatever is the complement of these nucleotides will come and stick itself to these particular to these nucleotides. Okay, and when they stick itself combined together, the T instead of T we will be having U, and this will just get stick to itself, and finally what we will have at the end, this new string of nucleotides, that is the mRNA. Now, this mRNA will be this thing, entire thing. This is called as a transcription process. This entire thing occurs within the nucleus. Okay, and the reason it occurs within the nucleus is because our DNA is inside the nucleus. So this entire process occurs within the nucleus. Then this mRNA comes out of the cell nucleus into the cell. Okay, so there is a cell, inside the cell there is this nucleus. So outside the it comes outside the nucleus to the cell. Okay, and here in the cell, remaining portion of the cell, which is called as the cell cytoplasm. Okay, here what happens is called as the process of translation. The translation process is basically to the major part, the second part of this, the central dogma principle, to form the protein. So what happens here is this mRNA goes and sticks itself to something called as a ribosome. Okay, so this ribosome, it again acts something similar to this RNA polymerase, acting as a starting point from where the sticking part will start. So to this mRNA, this transfer RNAs will come and stick themselves depending on what kind of nucleotides is there. Okay, so suppose we have UAA. So now it transfer RNA has like three nucleotides at the end. One end there is three nucleotides, and at the other end one amino acid, right? So these three are nucleotides which are at the end of this transfer RNA should cut and stick to this to that particular set of, to the first three nucleotides. Okay, then the same thing will happen to another to the next three. Okay, and we will be sliding along this messenger RNA. We just keep on sliding along this mRNA. We keep on sticking these tRNAs, and according to whatever nucleotides we are facing, three sets of three pairs of three nucleotides. Okay, combination of three nucleotides, we keep on sticking these tRNAs, and at the end, once the tRNAs are stuck, the amino acids will stick themselves to itself, forming a long chain. Now, these amino acids are released once we reach the stop part in this mRNA. When this entire mRNA has been read and we reach the stop part, then we will be stopping there. This set of three nucleotides is called as a codon. Okay, because it codes an information. We cannot have the entire set of like which each three of these nucleotides will have one particular amino acid, right? So which three nucleotides is referring to which amino acid, that is been coded. Which amino acid should come here, that has been coded into this sets of three nucleotides. That is why we call it as a codon. Okay, so these three sets of nucleotides are called codons, and for every codon, we will be sticking one tRNA, which will be having the complements of this set of codon in its one end, and on the other end, there will be an amino acid to this tRNA. So that amino acids will keep on sticking itself, forming the protein. Okay, that is the process of translation, and this is the entire uh, like this is what week one was based on. Okay, thanks.
Okay, ma'am.
Okay.
Okay, you maybe you can go through the content once. So maybe we can also take some part if you need in the next class if you have some
Mhm.
doubts and all. Just to confirm one thing. Okay. Sugani, can you stop the streaming?
Yeah, sure.
Okay. So, uh, You people got the calendar link and invitation?
Yes.